looking at this particular coronavirus, this coronavirus belongs to a family of viruses that are most closely related to the bat coronaviruses. There's no doubt about it. Most of the genome sequence tells us that this virus is definitely clustered within the bat coronaviruses. However, when we look at the rest of the genomic sequence, there's an insertion element. It's a very unusual element that actually is causing problems with placement of this virus. The Institute for Pure and Applied Knowledge analysis shows that the Wuhan strain of this virus clusters together overall. The published phylogenetics has a hard time placing it with only 75% support. And this inserted element, all of the other coronaviruses actually have 100% support, with one exception, that's 95. And so what's disrupting the phylogenetic signal of placing this into a unique monophyletic group, as it's called in phylogenetics, is, I believe, this inserted element. This inserted element, the origins of which was speculated by some Chinese researchers who published a study just last week, was speculated to have originated in snakes, but it's not based on its sequence itself. It's based on something called codon bias. When we encode proteins by DNA, different organisms actually have different biases in the codons that they use to make proteins from DNA. But that's not very strong evidence to say that that came from snakes at all. One team of researchers speculated. Since those researchers could not find any match, genetic match of that inserted element that we found at the N-terminus of the genome of this new coronavirus, nCoV 2019. Yeah, I've analyzed the entire genome sequence of this virus and compared it to the entire genome sequence of all the other coronaviruses that we have data for. And this weird element that doesn't belong there, I found that it actually did match a vector technology that was from the 1998 was the first time it was published in the Proceedings of the National Academy of Science. This vector technology is a mechanism by which molecular biologists insert new genes into viruses and bacteria. This vector technology is called a P-shuttle SN vector. Now, it's really unusual to find a vector technology sequence in a virus that's circulating in humans. And so naturally, one thing that we can say, I think for certain, is this particular virus has a laboratory origin. It should not be in the wild, animals anywhere. And in fact, if you then take that sequence and compare it to other proteins that it encodes, we find that it's actually a SARS protein that was put into a coronavirus for the purpose of making the vaccine work better. That's why this element, I believe, is in there. That's the science is showing us that that element is in there to create a more reactogenic, a more immunogenic vaccine. So if that's the case, that doesn't also rule out a bioweapons origin where they may do the same thing for other reasons. So if we could talk for a minute about the patents, of course, there's patents for every virus on the planet that's been genetically modified. The world is swarming. Laboratories are swarming with genetically modified viruses for research purposes, for the purpose of vaccines. Our country called for an end to the creation of these Frankenstein viruses that have more killing potential or are more contagious than their wild cousins in a very strongly stated article, specifically in 2015, that said, if we mess around with coronaviruses, we could get an accidental release. Now, the mechanism by which this particular virus made it into humans is unknown. Was it an escape from a bioweapons lab? We don't know. Was it somebody working in a laboratory that artificially, accidentally infected themselves? We don't know. Was it a vaccine that the Chinese government was experimenting with? And there's an interesting body of literature that shows that if you use a SARS vaccine in ferrets, in rats, or in monkeys, and then they get a secondary infection of a natural SARS virus, the elderly and the immunocompromised end up dying.